All right, guys, sorry about that. The video cut off on me. So the next thing is that I was going to do was to show you how the photosystem is arranged. So this is the photosystem here. Note that it's embedded inside of the membrane of the thylakoid. You can tell here's your membrane. And it's made up of all of these little discs, for lack of a better term. So each little disc is what's known as a pigment molecule. And see how they're all spread out? They're spread out that way so that all of them have a chance to grab energy from sun. It just depends on where does that beam of sunlight hit. So it can hit here, it can hit here, it can hit here. They're all going to do the same job. And that job is they're going to pass the energy, pass the energy, pass the energy until we get to a part of the photosystem known as the reaction center. So the reaction center is right here. In that reaction center, the energy is going to excite electrons that are just kind of sitting there waiting on it. We got the electrons from someplace else, and I'll tell you where later on. And when that energy comes in, those electrons get excited and they start moving. And when they move, they get moved from protein to protein to protein. And as they move, we are then going to create ATP. So that's what your photosystem is. Okay, so like I said, the photosystem is made up of all of these different pigments, those chlorophylls and the carotenoids that we talked about before. They're housed in these little areas that are known as light harvesting complexes or antenna complexes. And they are arranged the way they're arranged so that you can have maximum light absorption and then energy transfer. Okay? So in the reaction center, you have a pair of specialized chlorophylls that have proteins and are able to excite electrons. And they're going to use the energy from the sun to do the exciting, okay? Now, photosystem 1 and 2, because there are two of them, differ based on the characteristics of their antenna complexes and their reaction centers. So, in other words, the chlorophylls found in them are slightly different and they absorb in slightly different parts of the visible light spectrum. So that's why they're two different centers. Okay, so photosystem 1 and photosystem 2 are the two types of units in your photosystem, or in your thylakoid membrane. And they are directly involved in photosynthesis. This is where part 1, those light-dependent reactions, are taking place. Okay? So they're clusters or sets of pigments that are kept close to each other. And in photosystem 1, those pigments absorb in that 700 nanometer range. And in photosystem 2, they have an absorption peak in the 680 nanometer range. Now, it doesn't mean that they won't absorb in other areas, but it does mean that they won't absorb higher than 700 and higher than 680. So the way they're arranged, here is photosystem 2. So this is the one that's P680. And then here is photosystem 1, and this is the one that's P700. Okay, so what's going to happen, and here hopefully you would see something that looks very similar to a process we talked about in respiration. So I'm actually going to try and make this bigger so we can talk through this just a little bit. So we start off in that P680 photosystem. All these little green dots represent your pigment molecules, okay? So they don't show it here. Maybe they'll show it in a later picture. But water, well, they kind of show it, but they don't draw a picture. So the plant is absorbing water from the ground through its roots. The water is going to come in, and the first thing we're going to do is split the water into hydrogen and oxygen. So the oxygen that photosynthesis makes, this is where it comes from. Now, we don't need that oxygen, so the oxygen is going to leave, and we're going to work with just the hydrogens that are left. Now, hydrogen has one electron. we need electrons so that we can start moving things. So we're going to borrow those electrons from hydrogen. So the electron gets taken off of the hydrogen. It gets excited from the light energy. So that's what this little section is showing. And when it gets excited, note it moves. It goes higher in the complex. So that's how we know it got excited. So when it's down here, it's in low energy state or ground state. When it moves up, it's in high energy state or excited state. Now, as the electron has lots of energy, it's going to start moving from one complex to the next. So it moves down this protein, and then down this protein, and then down this protein, and down this protein. As it moves, it is losing energy, okay? The reason it's losing energy is because we're going to use some of its energy to move hydrogen ions into this space. So if you look up here, we have hydrogens that are on the outside. We're going to pump them across into the intermembrane space. This should sound a lot like the electron transport chain that we did in cellular respiration, because it's the electron transport chain, but for photosynthesis. So we start building up these hydrogens. Now, when we get to photosystem 2, our electron has lost its energy. So we're going to need to re-excite it. So here comes more sunlight. We're going to pass that energy until those electrons get excited. They're going to change state. And again, they're going to get passed one to the next to the next. Okay? They are being passed until they get to our electron carrier, which is NADP. So NADP is going to go ahead and bond with the hydrogen that we give its electron back to it, and it's going to make NADPH. So far we've made one of the two things. Remember, the light-independent reactions are supposed to make NADPH. The light-dependent reactions are supposed to make NADPH and ATP. Well, this is where our NADPH is made, but we haven't made any ATP yet, or have we? Remember our friend ATP synthase, that enzyme, that makes lots of ATPs? Well, here it is. Here's ATP synthase, and it's going to be the same process. All of those hydrogens are going to flow through ATP synthase, and as they do that, they are going to make ATP. So this is where the ATP and the NADPH comes from for the light-dependent reactions. Okay, so like I said, the process begins when your pigments, your chlorophyll and other pigments absorb light, and that happens in photosystem 2. So pigments absorb light in photosystem 2. Okay, now the light we're gonna pull the energy from the light and we're gonna transfer it to the reaction center in photosystem 2. This is gonna excite the electrons, and they are going to move because they're going to be accepted by a primary electron acceptor. Now, in the process, we're going to split water, and we're going to split water in a step known as photolysis. Okay? The reason we need to split the water is because the water has to donate its electrons to that photosystem so that they can be excited. Okay? Now, when the water breaks apart, it's going to release electrons. It's also going to release hydrogen protons, which we need to make ATP. And then it releases oxygen, which is a byproduct, and it's going to just leave the leaf. Now, once those electrons get to photosystem 1, they are going to need to be re-excited. And that happens in your photosystem 1. And then the electrons are going to get passed to an even higher electron acceptor molecule, which is your NADPH. Okay, so the excited electrons are moving, they're moving until they get to NADPH, and that's our final electron acceptor. This whole process we've just described is what is photophosphorylation. It's a lot like oxidative phosphorylation. So here's the difference. Oxidative phosphorylation happens in cellular respiration and requires oxygen to take place. Photophosphorylation happens in photosynthesis and requires light energy in order to take place. It doesn't need any oxygen. But the end result of the light-dependent reactions is the making of ATP and NADPH. Okay, so let's summarize real quick. So these are just the light-dependent reactions. Remember, they happen in the thylakoid membrane. We are going to need water, light, NADP, and ADP to start off with, because we're going to be making some stuff, so we need the raw materials. The results or the products are oxygen, ATP, and NADPH. The short version is energy is going to strike photosystem 2, which is your P680 peak. Photolysis of water is going to take place, which releases oxygen. Hydrogen and its electrons are separated from each other. The hydrogen is going to be pumped into the lumen while the electrons travel to the photosystem 1. As they travel, they are losing energy. When it gets to photosystem 1, it has lost all its energy, so another set of light is going to re-energize that, but that time it's happening in photosystem 1, which peaks at P700. Again, we're going to pass those electrons until we get to the final acceptor, which is NADP+. When the hydrogen and its electron go back together, NADP+ is going to accept that hydrogen, and it's going to become NADPH. While this is happening, hydrogens are flowing through the enzyme ATP synthase, and ATP synthase is going to convert ADP to ATP in a process known as photophosphorylation. So when we first started this, we talked about like cyclic and non-cyclic photophosphorylation. So it depends on the environmental conditions as to which one your organism is going to do. So here's the major difference. In non-cyclic, so if you think of cyclic as in cycle or circle, non-cyclic means no cycle. And a cycle has no beginning or end. So obviously in non-cyclic, we have a start and we have an end. So non-cyclic is what I've described to you, where we start off at P680, we get energy, we move, we get more energy, and then we make NADPH. And while this is happening, we're also making ATP. So the end result of non-cyclic photophosphorylation is we're going to make both ATP and NADPH. With cyclic photophosphorylation, we're actually going to circle back. So we're going to go through photosystem 2, and we will get to photosystem 1, but we don't have any NADP plus available. So when we get re-excited in photosystem 1, when we get re-excited in photosystem 1, we are going to send the electrons back to photosystem 2. So in non-cyclic, we go from P photosystem 2 to photosystem 1, and then finally to NADP+. That's the path of your electron. But in cyclic, we go from photosystem 2 to photosystem 1, and then we go back to photosystem 2. And it's normally because we don't have any NADP plus to pass the electrons to at the end. So in cyclic, we only make ATP, but in non-cyclic, we make both ATP and NADPH. Okay, so that was the light dependent. Let's talk about light independent. So your light independent reactions are also known as the Calvin cycle, and it's their job to store the energy that we've been capturing from sunlight in sugar. So there are going to be times when sunlight is not available. It could be stormy outside, it could be a cloudy day, or it could just be night. And the plant's still going to need access to energy. So when sunlight's not available, we are going to, when I say we, the organism that's photosynthetic is going to break down these carbohydrates that we've made in photosynthesis so that they can have energy to do all of the stuff that they need to do. Okay? So only some parts of the plant, of course, are producing this glucose, but the whole plant is going to need access to energy, so then it just gets moved around. Okay? Just because the leaves make it don't mean only the leaves use it. If the roots need it, then the leaves will send some sugar down to the roots so that they can have an energy source as well. Now, during Calvin's cycle, carbon dioxide is reduced. Remember, gain electrons, that's what reduce means, and then is converted into organic substances. We are going to need the NADPH because that's where our electrons and hydrogen are coming from. And then we're going to need ATP for the energy needed to do this. This kind of reaction only happens in certain types of plants. Those plants are called C3 plants. So plants that are able to use carbon dioxide and to reduce that carbon dioxide into sugar in the Calvin cycle, those are called C3 plants. So the first step of the Calvin cycle is called carbon fixation, and it involves taking CO2 and using an enzyme known as Rubisco. Rubisco stands for ribulose bisphosphate carboxylase. And we're going to use that enzyme to take the carbon off of the carbon dioxide and attach it to a holder molecule. Okay, so each turn of the Calvin cycle captures one carbon dioxide molecule. But if you think about it, glucose is C6. Well, one turn, one spin of the Calvin cycle gives me one carbon, which means that the carbon cycle has to spin 6 times to make a 6-carbon sugar such as glucose. Now, really what's going to happen is the Calvin cycle can make smaller sugars, and that's actually what's more efficient. So the Calvin cycle usually makes a 3-carbon sugar and then another 3-carbon sugar, and we put them together to make glucose. So there are 2 kinds of 3-carbon sugars. We can make PGA, which is 3-phosphoglycerate, or we can make G3P, which is glyceraldehyde 3-phosphate, but their claim to fame is they are 3-carbon sugars. Okay, and remember that a 3-carbon sugar plus a 3-carbon sugar equals a 6-carbon sugar, which is what glucose is. So if I did G3P plus G3P, I would get 1 glucose. So there are 3 steps in the Calvin cycle. So carbon fixation is the first one, where carbon dioxide is added to Rubisco, the enzyme. Carbon dioxide is added to RUBP, which is a molecule, and that molecule is going to be able to produce another kind of molecule that we'll use to make sugar from. Okay, so really what I need you to know is in carbon fixation, we are getting carbon from carbon dioxide. The next step is reduction, and in reduction, our NADPH and our ATP is going to convert our molecule to G3P. Okay, so in reduction, we're going to use the carbons that we got from carbon dioxide and the NADPH and the ATP to make G3P, which is a energy molecule. Okay, think of 1 G3P as half of a glucose molecule. And then the last step is regeneration. And in regeneration, the remaining G3P molecules are used to restart the process. Okay, so they're used to recreate our starting material so that we can do carbon fixation again. So, big picture. Where does the light-independent reactions happen? It happens in the stroma, so the liquid part of the chloroplast. The reactants are ATP, NADPH, and carbon dioxide. where you're going to produce glucose, ADP, and NADP plus. So our carbon dioxide is going to become a 3PGA, it should be PGA molecule. And that's going to become a G3P molecule, which will become a glucose molecule. The enzyme that's responsible for most of this is Rubisco. It fixes carbon dioxide into glucose. Okay, so really it takes the carbon out of the carbon dioxide so that it can be used to make glucose. Okay, so let's talk a little bit more about Rubisco. So Rubisco is an enzyme that is in charge of the conversion of RUBP, which is an intermediate, to fix carbon dioxide into G3P. And then it's further reduced to become glucose. So Rubisco provides the initial source of organic molecules for most of the world's organisms. Pretty much Rubisco is in charge of making all of the world's food, because without Rubisco we couldn't make sugar, okay? And then here it just tells us that lots of carbon dioxide is converted into carbohydrates every year, and it represents 50% or more of the total protein in leaves. So 50% of your total protein in your leaf is actually just this one enzyme, Rubisco. That's how much of it we have and we need. Okay, so additional photosynthetic products. If we have any extra of those sugar molecules, they can be used to make all of the other organic molecules. So other kinds of carbs, lipids, proteins, or nucleic acids. Usually we circulate photosynthetic products from cells to cells in the form of sucrose in plants. And then starch is used for your long-term store. So sucrose is everyday use, if you will. Short-term use. And then starch is how the plant stores energy for the long haul, for long-term use. All right, so again, this slide just goes over the two stages. It's the exact same one that we started off with. So you can read that on your own. And then this is the same schematic that we used before. So remember, we're going to have our light-dependent reactions that happen in the thylakoid. They're going to use water and the energy from sunlight to produce ATP and NADPH. We're also going to make some oxygen, which leaves because we don't need it. Once we have the ATP and the NADPH, carbon dioxide is going to come in and get fixed by the enzyme Rubisco. This happens in the light-independent reactions of the Calvin cycle, and that's taking place in the stroma of the chloroplast. Then we are going to rearrange that molecule using the electrons and the hydrogen from NADPH and the ATP that we've already made to make sugars. Okay, photorespiration. So photorespiration is a small problem that can happen to the enzyme Rubisco. So Rubisco can actually bind both carbon dioxide and oxygen. It should bind carbon dioxide for us to be able to make sugars, but it is capable of binding oxygen as well. So when the oxygen binds, we don't actually get to make sugar. We have two different processes that can happen. So if carbon dioxide binds to the active site of Rubisco, then it's all great. We do Calvin cycle, we make our sugar, everything's perfect. This is what we want to happen. But when oxygen binds to the active site of Rubisco, then we enter what's known as the photorespiration cycle, and we don't actually get to make any of our sugar, which is not what we want. Okay, so think of it as photorespiration isn't great because we're not getting our end result. We're not making our glucose. So how do plants deal with this? Well, remember, C3 plants are the ones that do the first method that I've described, okay, where we're actually getting into the Calvin cycle and we're actually making our sugar. But if if your enzyme is binding oxygen, you're not making sugar, so you need to do something different. So we have another set of plants that we call C4 plants because they found a way to deal with the fact that the enzyme can sometimes bind the wrong molecule. So C3 plants are called C3 plants because the Calvin cycle produces a 3-carbon intermediate product, which is that 3-PGA. Remember, that half of a glucose molecule.